Okay, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa. 안녕하세요. And hello all. Welcome and thank you, SynBYSS people, for joining us, the 168th seminar, and I call it a Cell Press Day. Last weekend on social media, I announced the list of 20 committee members for the second International SynBYSS conference scheduled from June 8 to 11, 2026 in Barcelona, Spain, including my co-chair, Marc Guer. I thank hundreds of the engineering biology community people for voting for the time and location, and the committee members for volunteering to help organize the conference. Thanks to the excitement and support of the SynBYSS speakers and audience, we already have more than 20 confirmed excellent speakers. We look forward to seeing many participants in Spain next year. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Dr. Emma E. She is a scientific editor at Cell, the high-impact premier biology and bioengineering journal, as you know. Last year, I met her for the first time in Hawaii, where the first International SynBYSS conference was held. As a journal panelist, she gave the participant valuable and excellent insight into publishing and the trend. I hope she and her colleagues will be joining again the second conference. In addition, as an editor, she supports the scientific society and helps shape the research field and direction. Emma, thanks so much for your contributions to the scientific community, and the virtual podium is all yours now, and thank you again.
Thank you so much for having me. I'm going to share my screen real quick. So I'm just going to give a pretty quick talk here, mainly just a rundown of just some, I guess, a peek behind the curtain or some tips, things that I find tend to help papers be successful. Just gonna hide that real quick. So yeah, hi everyone. My name is Emma Yee. I've been an editor at Cell for about 4.5 years now. And right, there you go. So Cell is one of many Cell Press journals. So here's a little sampling of them. We have primary research journals that publish in medical research, scientific research, physical sciences, that sort of a thing, as well as some trends journals that tend to publish mainly reviews and overviews of fields. And again, I work specifically at Cell. Every journal is editorially independent, but I work with other journals sometimes to facilitate transfers of papers and that sort of a thing. And a lot of the things I'm going to talk to you guys about today are relevant for any Cell Press journal. So you keep that in mind, regardless of where you're submitting your paper. They probably are also relevant for other journals, society journals, Springer Nature journals, and things like that. So yeah. Really briefly, what editors do. So scientific editors, particularly full-time editors like myself, editors who work at Cell Press journals, also Springer Nature journals and science journals, we are full-time editors and we are involved in every stage of the publication process for the most part. We interact with researchers and authors of papers. When papers are submitted to a journal, we make the decision whether or not to send the paper for peer review, if we think it's suitable for the journal, or to maybe suggest a different journal. We are in charge of peer review. We invite peer reviewers. We evaluate the comments that come in and make decisions based on that. And then we take the paper all the way, hopefully, to acceptance. So today, just really briefly, I wanted to share some tips that I have from my experience as an editor for different stages of trying to get your manuscript published. So I'll talk first about what things you can do before you've written the manuscript when you're just kind of early stage project development stage. Then when you're ready to submit a paper, you have it all written up and you're ready to go. And then some tips for after you get your reviews back and what things you can do. So when you're just kind of starting on a project, trying to think about, what experiments you should be doing, how you should be shaping this narrative, here's some tips that I have for that. So I would say something that is really important to do, and I'm sure that all of you do this, but I want to emphasize other reasons why this is important, is to do regular literature searches throughout the project. This is important for a variety of reasons, but also just to have a good macro sense of what is needed in your field and in adjacent fields. This is also kind of how editors think too. So it's always kind of good to be generally on top of literature in your field because that will give you a sense of, what are unanswered questions. Maybe what direction would it be fruitful for me to take this project in? Where's a space that not many other people are occupying, but like it's a needed question, and that might be a fruitful way to take the project. Also to think about what types of supporting data other similar papers have. This is helpful when you're planning a project that's maybe eventually going to turn into a manuscript because it helps you to early on think about like what types of data do I need to be collecting to set my manuscript up for success at maybe a particular journal that I'm targeting. So things like how much benchmarking is done. Do people in this topic space typically need in vivo validation? Is in vitro validation enough? Do I need to show a bunch of proof of concept applications, that sort of a thing? How do I show that something is functionally relevant, that sort of thing? And also just to have an idea of what types of journals you might want to target in the future. It's also good to do. Also, even if you don't have a manuscript written, it's really, really useful at this stage to just to talk to an editor if you can. If you're at a conference and there's a journal editor there, and you have some time and they have some time, you can just briefly go off and talk about the projects that you're working on in the lab, even if they're like 2, 3 years away from being published. It's, I think, always helpful to get feedback about what the editor at that particular journal is interested in, and the kind of data that they look for. Also, when you're maybe at a bit of a later stage, when you're starting to have a draft ready, asking other people to read your paper before you submit it to a journal, I think is really important. Can't tell you how many times when we get papers and reviewers and editors sometimes just don't understand what are supposed to be the key takeaways of a paper. And that's partially because I think sometimes people, when you write the paper, you've been working on this project for like 10 years even sometimes, and like you know what you're trying to say. But if it's not very clearly communicated, that can be missed by reviewers, it can be missed by an editor, and then that's not great for your paper. So trying to prevent that by having people who are kind of in the field but have not been working on the project as hard as you have for so many years, getting them to look at your paper is helpful, I think. So when you're ready to submit, again, talking to an editor is also a good thing to do to try to get a sense of what journal you might want to target. Searching for similar work at journals that you're thinking of targeting is a good idea to ensure that it's actually a good journal to submit to. So for example, how many papers on your topic has that journal published in the last like 2 or 3 years? If they've only published like one paper that's tangentially related to yours, it might not be the best journal to submit to. And also, again, looking at for that specific journal and similar papers, what kinds of like validation data they typically look for. There are a lot of journals will have like specific benchmarks. So for like certain types of topics, they need in vivo validation, or they need you to have some sort of benchmarking, like comparing to comparable tools and things like that. And so that's something that's important to look for when you're trying to decide which journal to submit to. You could also do a pre-submission inquiry. So not all journals do these, but it can't hurt to try, I think. So Cell in particular, we typically don't honor a lot of pre-submission inquiries. Usually if someone emails me, I'll just tell them to go ahead and submit like a normal paper. But a lot of journals do pre-submission inquiries. This is where you send an email to an editor. You can send it to the editor directly or to the general inbox for the journal, and you just kind of pitch them your paper. Usually kind of like a cover letter type thing. So you include things like what's the problem or the question you're trying to address, why it matters, what are like the key takeaways you want people to have from your paper, exciting aspects of the project, how it compares to other tools or how we currently think about some problem, and why you think it's a good fit for the journal. So kind of like a cover letter type thing. And the editor will give you feedback. They'll be like, oh, I don't think this is in our scope. Maybe you should submit somewhere else. Or they'll say, we're really interested. Like, we would definitely review this. Or they would say something like, yeah, we're not really sure. Like, maybe like send it in and we'll evaluate it. So this can be a good way to assess whether or not a journal would be interested in your paper before you actually submit. Um, yeah, when you send these, keep them short. We get a lot of emails. And if you want, it's optional. You could attach the full paper if you have it, or even just an abstract. Um, Tasek, feel free to cut me off if I'm going too long. I'm not used to 5 to 10 minute presentations, so if I'm good, okay, sounds good. All right. Yeah, and then I think this is also something to keep in mind too when you're either sending a pre-submission inquiry or just starting to write your manuscript. This is especially, I think, relevant for I would say like maybe higher impact factor journals or journals that are looking for something that's a big advance over what is currently done, something that's like quite distinct from what we're currently thinking. So if you're targeting one of those journals, thinking about like how the editor is going to be thinking about whether your paper is suitable for that journal. A lot of it has to do with how much of an advance I would say that it provides to the field. Um, Novelty isn't necessarily all that these journals are looking for. They're not just looking to see, like, is this the first time anyone has ever done exactly what you've done? A lot of times they're thinking about things like how many people are impacted by these findings, and how many people are going to be interested in these findings. So if you have like a genetic circuit or something, like how many people are going to want to use that? How many different fields is that circuit applicable for? That sort of thing. Because these are things that are important to put in like a pre-submission inquiry or your cover letter. Speaking of cover letters, yeah, so I think I'll just briefly go through this because a cover letter, much like a pre-submission inquiry, you want to include kind of these key things. What's the problem? What did you do? What are the, key takeaways? Just to make sure that the editor who's reading this cover letter is in the right mindset when they're going to read your full paper. Usually the cover letter is the kind of the first thing an editor sees too. So you want to kind of make sure they're in the right frame of mind, they understand what you did, they understand what they're supposed to take away from this manuscript. It's important. In the cover letter, I actually think it's really useful, to me at least, to mention similar papers that have been published in that journal. So I will admit that there are times when I'm kind of on the fence about a paper and I'm like, oh, I don't really know if this is suitable for Cell. It seems like maybe it'd be for a more specialized journal, but the author was really good about saying like, hey, you guys published like X, Y, and Z last year. It's really similar to my paper in these ways, but like, what we do is like, distinct in this way. Or they'll say even things like with competitor journals or something, they'll say like, oh, Nature also published something in this space that's related and that sort of thing. And I think personally that if an editor is on the fence about your paper and you have some like compelling evidence that the journal or similar journals have published stuff in that space, it could potentially help the editor decide to send something for review. So, and on that vein too, I think this is important both for reviewers and also for editors, is to in your introduction or your discussion to think about like what are people going to compare your work to? Because I think reviewers and editors do this all the time, is like they read a paper and it makes them think of something that they've read recently. And they will automatically compare what you're doing to what was previously done. And so it's good to get ahead of that and think about here's all, these things that someone might think of. These are like comparable work. And let me explain why what I'm doing is different or is an improvement on what's previously been done. Because it's better for you to explicitly explain that than to depend on the editor or the reviewer to like figure it out themselves. So I'd recommend doing that in the cover letter, but also in your paper as well. Um, Sorry, I don't want to take too long. But yeah, I think the abstract is also something that's important to think about. And the main thing that I wanted to say on this slide here is that something I think people don't realize is that when I invite reviewers, the abstract is the first thing that they see. That's all I share with them. I'm like, would you be interested in reviewing this paper? I think your expertise would be good. And here's just a little snippet. Would you be interested? And if you have a good abstract, you're way more likely to get reviewers to agree to your paper. It's easier for me, then I don't have to invite fewer reviewers, but it's also easier for you because then your paper isn't languishing in peer review for a long time because it's a lot easier to get reviewers signed on and then they will hopefully submit their comments on time. So I think an abstract is good for many reasons, but for speeding up peer review is also a reason to have it. Um, yeah. Also, when you submit for cell press journals, at least, I'm not sure about other families, but you don't need to worry about word count. Don't worry about formatting. Just, you can send in a Word doc, you can send in a LaTeX format. As long as it's visible, things are logically laid out for reviewers, that's really all that matters. You don't need to worry about any of that stuff. Just submit it. Preprints are fine. Preprints are great. We love preprints. It won't impact the outcomes of your paper. And also suggest reviewers too. I know that recently, I think some Elsevier journals have discontinued this, but Cell still allows this. I think there are still some other Cell Press journals that allow this. Suggested reviewers are great. It's a hard time to get reviewers in the last few years. You will, again, help speed up your own peer review process and also help the editor. But do remember to try not to suggest people you have like conflicts of interest with and things like that. Um, all right, I can try to speed through this pretty quick. After peer review, editors are they're making decisions based on what reviewers say. And keep in mind that you can discuss revisions with an editor. So, for example, if you get your reviews back, um, regardless of if it was a rejection or if it was a major revisions decision, if the reviewers are asking for things that are just like literally impossible, or they would take 5 years to do, or something like that, or they're just not super relevant to the study. Like maybe the reviewer is maybe not quite understanding what the main applications were, or they're like kind of going off on their own personal tangent, asking you to do something that will satisfy their own personal curiosity. Tell the editor that because we try to be aware of these things, and I'll read through the reviews and be like, oh, that sounds like they're asking them to build a whole new mouse model and that's silly. But sometimes I miss it. Especially in broad journals where the editor is covering a lot of different topics. It's important to make sure that if any of this stuff is there, to tell the editor that this is why I don't think that we can address this comment, and it can be a conversation. Also, editors can clarify reviewer comments or request feedback on your behalf. So if a reviewer has a comment and they're like, you just don't really know what they're asking, or they're being really vague, and you want to address it, but you have no idea what they're saying, just email the editor and say like, hey, reviewer comment number 5, like I'm not really sure what they're asking for. Could you clarify? And then the editor can do that. And that will, I think, can only help. You also have the option to appeal decisions too. It's something to remember. If your paper is rejected at any journal, you can send the editor an email or send it to the general inbox. I'll try not to take too long here, but basically, it's a good idea to appeal if you have data or you know that you can address basically every single one of the comments that the reviewers have made, or all the main ones, or if the reviewers were asking for things that are just not possible. If the editor has said in their decision letter, rejecting it for X reason, and you have data that can, solve that problem, that's a reason to appeal. It's not as good of an idea to appeal if there's kind of like subjective, I guess, disagreements on extent of novelty or advance. These are things that are hard to resolve. Sometimes reviewers will think a certain thing, and it's hard even with additional data sometimes to overcome reviewers saying that that this isn't something that's suitable for the journal. Or if it's undergone a lot of peer review rounds, I found that with every additional round of peer review, the chances of convincing the reviewers otherwise diminish a lot. And it's not always the most constructive to consider appealing when it's at that stage. And then you also have the option to transfer at Cell Press journals. If you're rejected, you can transfer to another journal, and all of your stuff can be moved. You can have the option of using the same reviewers. That can speed up the process. That you can go to another journal and you don't have to start from scratch again with peer review. That's basically it. I have a few more slides, but I don't think that they're super necessary. So I can stop there, or I'll go all the way to the end if there's questions. Yeah.
Thank you so much. That's very valuable information. something I wish I knew when I was junior faculty or postdoc or even earlier. Now I do everything, including sometimes, rebirth, and then actually we are actually revising that one because now I realize editor wanted to help authors, and then editor super sad when they reject the paper. I didn't know that one until I became a editor. So because the editor wanted to shape the community and then helping author, and then their worst part of the job is rejecting paper. So if you are unhappy with your decision, talk to editor, and then that is actually one thing I also want to emphasize one more time because I was shy, I was afraid to talk to editor, but that's not the case. They want to help you.
Definitely.
Yeah. Thank you so much. That's absolutely wonderful presentation. All right. Let's move on. Okay, now the main speaker of today with a longer introduction. Dr. Miguel Jimenez is an assistant professor at Boston University, where he runs a microbial interaction group. The group integrates engineered microorganisms with mechanical and electronic devices for applications in human health, agriculture, the environment, and also entertainment. He received his bachelor degree in chemistry and chemical biology at Harvard University under the supervision of Damian Young and Stuart Schreiber, and also received his PhD in chemistry at Columbia University under the supervision of Virginia Cornish. He completed his postdoctoral work with Robert Langer's lab at MIT, a pioneer speaker in this series as well, and also I believe the most influential bioengineer in history. And also most recently, Miguel was a research scientist with Giovanni Traverso at MIT as well. It is kind of coincidence today, me, Miguel, and Emma all, went to MIT, and I also probably also need to say, I mean, today is the MIT day. So Miguel, thank you so much, for your time today. Please take it away, all yours now. Thank you.
Thank you so much, Tessa. Really excited for the invitation and the forum here to get to share a little bit of my work and a little bit of what our group is doing at Boston University. So I think if anything, if you get anything out of it today, I just want you to learn this idea of microbial devices, as Tessa introduced, really the idea of designing systems that incorporate microbes as a component in them, but really rather than just doing genetic engineering, really we're doing systems engineering of the microbe, the mechanical aspects, the material aspects, and the electronic aspects for a bunch of different applications. If you'd like to follow our group, I encourage you to look at our LinkedIn page or our group website listed here. And again, thank you so much for the opportunity to share our work. Okay, so as you may well know here, in this community, particularly if you're in the community of synthetic biology, you might know that microorganisms are really all around us in forests and animals. And as I list here on the right side, they're incredibly important and have been important for food production for society. What I'd like you to observe here from all these different pictures, though, is that microorganisms really live in a very diverse set of contexts. For example, the soil, the gut of a cow, the rind of a cheese, really complex environments. However, by and large, the way that we've studied them in scientifically and even industrially have been in very idealized settings such as fermenters, shake flasks, or petri dishes, which are really a narrow set of conditions contrasted with the incredibly large set of conditions that organisms actually see in their lives. And there are now advances on that. For example, microfluidic advances and even high throughput culturing. They're the newest ways of sort of looking and understanding microorganisms. But my contention today is really that these are just smaller ways of doing the same sort of large shake flask experiments. And there's really no way of looking at organisms in really these complex environments or leveraging the capacity they have to function in extremely complex environments like we see in nature. And so our group attempts to leverage this versatility of microorganisms to use them in new ways and integrate them into better working systems. So today, I would like to tell you two stories. One on the left side, we'll start with microbial materials used for therapy. And the second story will be really this idea of microbial devices, in this case, uses diagnostics. So I'll jump right in. So here, this story was recently published. We developed synthetic extremophiles, or really dry stabilized microbial therapies. And this was work funded by NASA and DARPA. So as you may or may not know, microorganisms are being developed as drugs. So here you can see there's a range of different diseases that microorganisms are being applied to. These are actually in clinical trials in humans, really industry-led clinical trials where the actual active pharmaceutical ingredient in the clinical trial is a living microorganism. You can see a large proportion applied to infectious disease, but really the spectrum is really quite large, including diseases such as autism as well. What we can see, though, when we look at these trials is that the way that these microorganisms are being administered to patients is through either rectal enemas or frozen drinks. So while this has enabled the capacity of these companies to test whether these microbes are effective or not, it really limits the scope of how these microbes might be able to be used. For example, imagine that you needed to use one of these administration routes if you just had a headache, right? And so we see that there's a challenge here in the field. We need to be able to move on to different ways of administering microorganisms if they're really going to be this sort of next generation of medicines. You might say, Miguel, I see lots of microorganisms on shelves. For example, probiotics are sold in pharmacies. They already come as tablets. Maybe some of you watching take probiotics. But what happens is that we actually went and looked at these. And if you look at very closely on the boxes, first of all, these are food supplements. So they're not actually tested to treat a specific disease mechanism. And if you look closely on the boxes, they often say that while they might have had live microorganisms at the time of manufacturing, they're not guaranteeing any activity or even viability of these organisms in these tablets. We wanted to be sure of that, so we actually did this small survey of commercially available probiotic tablets, which are really the only commercial example of microorganisms dosed orally. And you can see here that by and large, these organisms, these commercial products have much lower viabilities than promised. And in the cases where they do sort of have the amount of promised cells, the way they're doing that is by having an extremely large number of cells that are mostly all dead. And so only a very small percent of those cells are actually alive. What we also saw here is that most of these products are these gram-positive microorganisms. And for example, E. coli, which is one of the workhorses of academic synthetic biology and some up-and-coming companies, which is represented here in blue, Mutaflor, was one of the least stable organisms, really having very low viabilities for E. coli in these dry products. And so why is that a problem? So if we go back and think about the clinical trials that have been run so far, where you have these active ingredients that are living cells, you can have companies like Seres and Synlogic, you can look at the amount of organisms dosed in those trials. So they range from 10 to the 8 to 10 to the 12 organisms per dose per day, with an average of around 10 to the 10 to 10 to 11. So if we take the viability that we found in the commercial products and imagine having to use the commercial product to reach these doses, we can see that if we make that calculation, we would have to be taking 10,000 pills a day or more. So we're just like orders of magnitude away from being actually able to have orally administrable organisms, and that's why companies choose to use rectal enemas or these other sort of large frozen drinks that need to be cryo-cooled. So with this problem, we really thought, okay, how can we improve on this or fill this gap? And so we thought, okay, yes, we would like to have dried organisms, but drying organisms has been around for quite a while, and there's lots of sort of freeze drying stabilization work that has happened before. However, when we really thought about it, the way that it's been done previously is really in this sort of piecemeal way, and really the primary application so far has been for stabilizing microorganisms for culture collections, where the goal is just to get just enough organisms back when you rehydrate them so you can grow them again, as opposed to this new use of microorganisms as living medicines, where you want a really high dose immediately after ingestion or immediately after hydration. And so because of this, we decided to take this unbiased approach to searching the space of material strain combinations. And so on the left side, we set up this search with 4 different organisms, E. coli, Nissl 1917, and S. boulardii, which are 2 of the sort of top synthetic biology organisms that are being used as medicines. And then 2 comparators, L. plantarum, which is an example of one of the gram-positive organisms that we see in probiotic products, and then E. mellilati, which is another example of a gram-negative, but it's actually used in seed coating. So we just wanted to, that was another example of organisms being used in a dry state commercially. On the right side, we decided to limit the materials we looked at to GRAS compounds, or generally recognized as safe compounds, so that at the end, anything that we would find could be immediately translated into the clinic, because we wouldn't need to have that sort of material approval of some new synthetic material. What we developed was this high throughput viability assay using batch freeze drying on shelf lyophilizers in microtiter plates. And then for a while, we were doing viability assessments using sort of these available kits, but we ultimately found that those assays led to a lot of false positives. They didn't really directly measure whether the organisms were alive or not, and often had cross reactions with the materials we were looking at. So instead, we developed this direct plating approach where we could plate in batch and programmatically count colonies to be able to look at thousands of results. When we look at the results from that large survey, which we see here, we can see the 4 organisms in the rows. Each column is a different material that we've been looking at. And then the 2 sort of sets above is like a higher concentration of material and a lower concentration. And so we're able to see that, in fact, we see hits, we see differences. We're very excited that this approach and platform actually works. Circled in those black boxes, we see that some materials that are previously established to be stabilizers, such as trehalose, work, but interestingly, they weren't sort of the top materials, and certainly not the top across the board. So we're able to recover these known compounds in this survey, but are able to get a lot of information across a lot of different other new combinations of microorganisms and the material. When we analyze these results, we can bin each of the materials into these different material classes or chemical classes. And we can see that there's an overrepresentation of cell extracts and saccharides, as well as peptones in the top performing materials. This also recapitulates some established results in the literature previously. But again, previously we only sort of had that idea in a piecemeal way. And here we were able to really map the entire landscape of those different compounds. One very interesting result we got from this data is that, we were expecting that potentially there would be like 1 or 2 materials that worked really well across the board. But what we see is that there's no silver bullet. It seems that for the very, very high or very top performing materials, there seems to be sort of a species specific combination, which is much better than the kind of like a general working material, which is very interesting. We, of course, after the survey, we revalidated those hits in a traditional way, just using large scale lyophilization and plating. But here we started asking, which of these materials that are all top performing, how do we differentiate them into better versus less good materials? So we did also this 30-day stability study at room temperature. And so you can see that even though a lot of these materials work very well after just 1 day of storage at room temperature. We started getting differences after 30 days, as you can see here in red. And moving beyond this, using these platform allows us to then sort of further optimize these materials. So we decided to focus on E. coli Nissl, again, because we saw that there was the biggest gap there in terms of its sort of intrinsic stability as we saw in the commercial screen. And so now we're able to do combinations of materials. So not just look at one microbe and one material, but look at two material combinations and also combinations of concentrations of those materials. And so for E. coli Nissl, we were able to do that, map that space really quite efficiently using that approach that I introduced to you. And we were able to see here that the top performing material for E. coli Nissl was SynBYSS, but then in these other combinations, we're able to get other formulations which return formulation D and E, which are even better performing in this more stringent test where we keep them at elevated temperature, so not just room temperature, but 37 degrees for up to 23 days. And so sort of to conclude what we can do with this new material, or to contrast it with the available product, we can see here in a head-to-head assay between Mutaflor, which is, again, the commercially available product that incorporates E. coli Nissl. So this is, we open up those capsules, test their viability. When we compared with the single material, SynBYSS, and two material combinations, formulation D, we see that after 1 month at room temperature, our approach yielded new materials that improve the viability of this microorganism in the dry state by over 3 orders of magnitude. So that was super exciting. And then we decided to take this even further out to over 6 months of stability at an elevated temperature. So this could be considered an accelerated stability assay. And we can see that we now start seeing differences between the two material and single material formulations here, where we see formulation D in green has lost just 1 log of viability after greater than 6 months. This is in contrast to the blue line, where we formulated coli Nissl with maltodextrin, which is the stabilizer found in the commercial product Mutaflor. We can see that after just 2 weeks, we lose all measurable viability. So really sort of shifting the slope of that curve significantly, which sort of gives credence to this approach of finding stabilizers for microorganisms. So what, right? So now we have these more stable dry powders. What we were particularly excited about is now having stabilized a microorganism as a powder, we saw the possibility to plug it into existing manufacturing processes for pharmaceuticals. So, when you take a medicine, it often isn't just a powder. It might be a tablet. Like you, for example, can think of Advil. It could be a tablet that then is coated with a candy coating, for example. And all those processes allow us to modify and improve the efficacy of medicines. And so we wanted to see if this approach allowed us to do that. So what we did here is we stepped through these standard manufacturing processes, such as milling the powder, mixing, compression, tableting, coating the material. And so we were able to see that when we can contrast sort of the existing commercial approach in red with our improved formulation, we're able to get significantly higher viability through these very harsh processes that involve high heat, exposure to organic solvents. So this is very exciting because it really allows some of these powders to be used in a sort of plug and play manner, or just drop in replacements for other powders that are already available to the industry to make these advanced medicines and dosage forms. One particular thing that we were interested in is the controlled release of medicines. So there's a well-established matrix former, HPMC, that when mixed into tableting formulations and pressed, it slows the release of usually small molecule medicines. And we were able to demonstrate that when we measure sort of an activity from these cells, so luminescence, so the cells need to actually be metabolically active, we're able to delay the release of this activity from these tablets. And again, this is enabled by the fact that we have a very high viability powder to begin with, and therefore are able to formulate it in the dry state with these matrix formers and control the release from these tablets. So you might imagine that this might enable or open the door for developing medicines that don't just have a simple dry organism in the tablet, but might have 2 or 3 or 4 organisms altogether being released at different times, tuned to develop a specific treatment effect using this new type of medicine. So this was very exciting work. And in our current group now, we're envisioning that with having this ability to stabilize microorganisms, we can actually use them not just as medicines, which is super exciting, but also start using them as sort of solid materials or enable manufacturing processes that allow us to integrate them into devices. And so with that, I want to transition to the second story for today, where I want to tell you how we're using microorganisms as these sensor systems in a diagnostic ingestible electronic pill, and sort of the process we had to do to get there. Okay, so this development of an inflammable bowel disease sensor was really with collaboration with Maria Henia and Mandy Liu from the Liu Lab and the Yasisigi Lab at BU on synthetic biology and electronics. This is work funded by the Helmsley Foundation for IBD. So as you may know, IBD is a class of diseases usually split up into Crohn's disease and ulcerative colitis. This is a disease of chronic inflammation of the intestine. And the way that it is monitored and assessed is really through these invasive and slow procedures. So most often colonoscopies, CT scans. And while there are sort of less invasive tests, those available are really blood tests or stool tests, which are still very slow and sort of take days to weeks to get those results back. So we wanted to ask, is there an ability to actually have something such as like a Fitbit you can swallow that can monitor your inflammation in this sort of chronic way that might allow you to understand what aspects of your lifestyle are maybe leading to flare-ups and allow a better ability for patients to monitor this chronic disease, as well as their doctors. So others have imagined using microorganisms as diagnostics previously. For example, here from the Siller Lab, as well as iGEM teams, we have these different organisms that are responsive as they pass through the gut. Something you'll notice, though, is that a lot of the time the readouts are simply not tractable as commercial products. For example, there's still these academic readouts, for example, counting colonies, or these engaging readouts such as color changing poop, but really that makes it hard for patients to really act on that information. And so our idea here, or our vision, is really, can we offload the readout to a wireless electronic? This is the idea that we're really sort of dividing the labor into the biological part, allowing it to do what it does best. So for example, chemically specific sensing can be done by proteins in the organism, and then the computation and wireless communication can be done with electronics, which are very great at doing that and plugging into our existing personal devices. And so that combination here on the left side, we had used synthetic biology tools to engineer E. coli Nissl to detect 4 markers of inflammation, or 4 mediators of inflammation, as listed here: nitric oxide, tetrathionate, hydrogen peroxide, and thiosulfate. These are reactive chemical species which mediate inflammation and which cannot be measured today in the fecal matter because they react away by the time they exit the body. And on the right side, our collaborator Rabia Yasisigi in Boston University's electrical engineering department developed an ultra low power photosensor chip that allows the electronics to quantify the amount of light generated as a result of detection by the organisms. So these organisms can bioluminesce and report on this detection event. And so really our work in this space and what we continue to do in our research group at BU is really this integration piece. So we sort of are taking these off-the-shelf synthetic biology circuits and working with these advanced up-and-coming microelectronic systems and asking the question, how do we marry these two worlds, the wet world of microorganisms with the dry world of electronics, in a way that allows the full system to work efficiently? And here the idea is that you can have a pill that's really small enough to swallow, and as it transits to your gut, it is wirelessly reporting on inflammation to your personal device and allowing you to track those levels. So there has been, the field of ingestible electronics has many advances recently. And so here, for example, you have the idea of having an ingestible camera. This is a commercial product pill cam that allows video endoscopy. And even the idea of including some bacteria in electronics was previously done by our group, our previous group as well. One thing you'll notice though with these devices is that while the people have demonstrated or even have commercial examples of ingestible electronics, the devices are really quite big. And so this is a really important, as I learned through this project, really, if you're asking questions, can we really translate this? Can we really sell this and put it in the clinic and have patients actually use it? We really need to think beyond just the basic science mechanism of the device function, but also understand any safety concerns with this. So with ingestible devices, when they're very large, can really cause obstruction, which is a medical emergency. So if you were to swallow these pills and it caused an obstruction, you need to immediately go to the emergency room to have them often surgically removed, or else you might die. So when we compare the sizes of these devices, so on the left side, you have PillCam, which I just showed you, compared to the sort of older version of an ingestible microbial device. And you can see that they're really quite big. And so PillCam is actually a commercial product. So theoretically, it should be safe. But even this product at this size scale leads to about 1.4% retention or obstruction rate in patients who consume these cameras during video endoscopy procedures. So because of this product is actually not safe and is not allowed to be taken at home. So PillCam, while very effective at its function of recording its transit through the GI tract, has to be consumed by the patient under the supervision of a gastroenterologist in their office because of this obstruction rate. In contrast to this, we can see in the commercial side that there's are non-dissolvable dosage forms such as Procardia XL. These are medicines, so there's no electronics in them, but they're basically tablets that are plastic that you swallow, and they don't dissolve. And so we were able to see that if you sort of cross this threshold and have a solid object that is this size, all of a sudden you eliminate basically all obstructions. And so this medicine has been dosed over 4.4 billion times, and there has only been 85 cases of obstruction, which means that it can be safely taken at home. So that was really our goal with this integration challenge. Really, can we integrate these electronics with these organisms in a small enough package, basically around the size of a blueberry, so that it can be safe and yet retain the function we're after. So some of the technical challenges, for example, is if we're able to only load 1 microliter of cells, can we get enough signal from these cells that's high enough to be measured by the electronics at a low enough power? Also, how do we contain those cells so they don't exit the device, and how do we protect the electronics from ingress of the liquid while allowing ingress of the analytes into the cell compartment? And so to sort of cut a long story short, what we were able to design after many trials of different types of designs was this unibody pill design, where we have a plastic capsule that sort of a clamshell design around the electronics, but which directly incorporates these small microwells that hold about 1 microliter of liquid. At this size scale, you really can't use screws or compression gaskets, and so we use instead these pressure sensitive adhesive films, both to seal the device overall, as well as to seal filter membranes onto this wells. So from the outside, you have this PSA film sealing off a permeable membrane to allow analytes into the cell compartment. And from the inside, the backside, we have an optically clear film also with a pressure sensitive adhesive that can seal the inside. And so you can see that when we look at these chambers from the inside, we can see that we can distinctly load them with these different color solutions just to represent the microorganisms. And then from the outside, you're able to see that these solutions which can be replaced with the microbial solutions, are retained in their specific spots. So it was very exciting when we finally were able to get this encapsulation system working. And again, when we put everything together, when we actually load these genetically engineered microorganisms into these wells, we're able to detect a significant increase in luminescence via those custom chips. You can see here that upon exposure to tetrathionate as compared to vehicle control or a buffer not containing tetrathionate, we see a large increase in the photocurrent measured by Rabia's team's custom microelectronic chips. So this was very exciting. And because we have multiple wells, you can imagine that we can use load different sensors as well as control sensors that allow us to normalize the signal, both the biological signal as well as the electronic signal to measure the dark current of that electronic. We wanted to go beyond this and really ask, can this integrated system work in a true disease context? And so we used a large animal model, Yorkshire pigs. So these are large, 60 kilogram pigs where we're able to place devices that are the full size for human scale consumption, so about the size of a blueberry, into this intestinal segment system. So these animals are alive, they're under anesthesia, we're able to place the devices into the intestine, close them back up, and ask questions about like, how long does it take, or how much signal are we actually getting wirelessly from the device through the full thickness of the animal in the actual context of the intestinal lumen. So now the device is being exposed to all these intestinal juices and has to continue to work as expected. And so very successfully, or we were very happy when we saw these results that in this full living system in a Yorkshire pig, again, the device is able to detect tetrathionate and read out that information wirelessly from a handheld device external to the animal. You can see that there's still things to figure out here in the sense that we're seeing sort of this decrease in signal in the buffer control, but we're still able to see this large delta between the two signals, which indicates the presence of tetrathionate. So very exciting. And just as a bonus story, I just wanted to mention a little something about what we're working on at BU today. So we're sort of taking those different baseline advances in terms of stabilization, integration, demonstrating the ability to integrate devices into microorganisms into devices, and thinking about what are all the other places where we might be able to do this. And so we started this group at BU in January of 2024 of last year, about a year and a half ago. And we had this really great master student, Kavya, and our undergraduate, Luca, who really teamed up together to think about how we might use microorganisms for fertility hormone detection at home. Through some interviews and collaborations with medical doctors at BU's medical school, we were able to really understand that while we might think to detect fertility hormones in urine, something that might be easy at home, really doctors want to know the quantities in blood. So we imagined, again, thinking, can we leverage microorganisms to really work in these complex systems, and will they actually be able to give a signal? The reason there is because, as you might know, during pregnancy or assisted reproductive treatments, in collaboration with Dr. Wendy Kuhong, we understood that there's this large burden placed on women to track their hormones. So often having to take time out of their day to go multiple times in a week to get blood drawn and assessed in the hospital's pathology lab, and really other available at-home monitoring tests, such as these p-sticks, don't really provide a quantitative enough signal to enable the treatments. And so just as a small snippet of data, we're able to see that whole cells are actually able to detect estradiol, which is one fertility hormone of interest. And what I would like to highlight here, though, is that this detection that we're able to see here, read out by fluorescence, so not yet something that can be directly integrated into the microbial device that I presented previously, but very promising, is that we're getting the signal in whole blood. So this is in undiluted blood with nothing else in it, just adding the cells. we're able to get this signal, really showing the versatility of whole cells to function in these complex environments. So we're very excited about these results. And with that, I just want to thank all the people who made all this work possible, both the people and the sponsors here for the work presented today, in particular the Helmsley Foundation, Trish, NASA, and DARPA. And with that, I just want to mention a little bit about our group. We're at this budding group at BU. On the left side, you can check out our website. And we're also host the annual BU iGEM team. And so most recently, the team developed this AgriNova and Nova device where, again, we're integrating microorganisms, in this case, for detection of agriculturally relevant compounds. And so I just want to leave you with that, hopefully the takeaway here is that I've convinced you that microorganisms can be used for quite a lot of things. And we're sort of moving beyond these initial examples in synthetic biology of just demonstrating function in the lab and really trying to solve some of the challenges that are stopping them to be applied in the real world. Thank you.
Thank you. This is exciting talk and fantastic. Especially, Maria, your collaborator, actually covered, I think, before the publication, I think the genetic circuit part, and then you now cover the rest of, part she actually didn't cover at the time. So I now have full picture of the entire research, even though I read the paper. Excellent. I want to ask Emma whether she has comment or question and anything.
Not really on anything specific. Maybe independently, I would love to hear more about the your new hormone whole blood project, but I think it sounds really interesting.
Thank you. Yeah, we're very excited. And again, as I mentioned previously, that's one of the things we see as super important to apply it to. And we're also looking at other diseases as well, sort of continuation of the IBD project. Recently, we got some funding from the Kenneth Rainin Foundation to now look at a therapy angle on using these microorganisms in that context.
Emma, do you have any other comment or question?
I don't think right now, no.
Sure. In that case, let me ask one technical question about the first part of your talk. So when you show the viability data, I'll be a bit disappointed because I'm also using probiotic for the drug delivery or the also the diagnostic tool, and I cannot now start to understand why SynLogic couldn't deliver the product. That's a very disappointment to me. So some technical question about that. When you do that test, did you just directly kind of take the pill and then put it into the whatever media and then test the viability? That's how you did.
So we didn't test the dosage forms. We tested the what you would call the API, the active ingredient. So anytime that there was a capsule, we opened the capsule, took the powder out, and rehydrated the powder. In the case that they were tablets, we crushed the tablet the way that you might have that disintegrate in the stomach. So again, we were trying to assess really the microbial ingredient in it rather than the dosage form, which might be the outer capsule.
But did you do also some test mimicking the real, kind of scenario, going through the very acidic, condition, etc.
Mm-hmm.
Did you do that, or also
right. So for the probiotic, the commercial survey, we didn't. Those where we were just looking at the viability of just hydrated in water. So we didn't have a more stringent test, but even in this sort of less stringent test, it already was showing these poor viabilities. For our scenario, we did. So that was actually something that through the editorial and review process,
Mm-hmm.
we were encouraged to amplify that work. So I don't present the data here, but if you read the paper, you can see here that one of the things we can do is we can enteric coat these tablets. So we can have the powders, press them, and then coat the tablet. So normally this requires, again, higher temperatures, organic solvents. But we were able to have this improved formulation, which is not actually on this graph. It's in purple in the paper. But even for this stage, is has even higher viability, up to 10%. And once you put an entire coating, that works just as designed. And
Mm.
so we're able to now place it in a low pH environment and then in like an intestinal environment and in a neutral pH environment. And we're able to see that viability is retained as expected. And
Mm.
so again, the idea here is like we don't have to reinvent the wheel for low pH protection. We just have to sort of solve this problem of having a high quality, very high viability powder, and we can drop it into this existing technology of enteric coating, which is really, there's lots of different things you can do there that's already available commercially.
So that's exciting. Even though I mean the capsule protect the probiotic inside, but some die during the process of the going through the stomach. But with the new, material, I guess, I mean, a lot of, new kind of approach could be, possible. And I hope, I mean, the SynLogic did test all of that and whether
All right.
that is possible or not, because I was so excited when they talking about phase 3 clinical trial, but I don't know why they dropped the drop. Um,
Yeah, no, I mean, I was also following that company quite closely. Yeah, my understanding is that, I mean, something that they really sort of were trailblazers in is just showing just how many microorganisms you can dose to patients and have it be safe. And so I think those results still stand. But I do think, this is a personal opinion, that some of the poor results in the later trials were due to dosing.
mm,
And so, something that the field is starting to think about now is not just high viability, but high function of the thing you're asking about. So here we're like simply measuring viability in this graph, which is, yeah, step one. But what we're doing here in this sort of a little bit more uh, complex step is that we're actually measuring luminescence as a model pathway that's a biosynthetic pathway for, a photon, and really measuring how quickly can you regenerate that product after hydration, drying, and then rehydration. And so I think that's kind of a next step that's going to be super important for those kinds of companies, is to look at actual stabilization of function, not just viability. So I wonder, yeah, if they retested everything now with this hindsight, if there's newer companies that will be able to cross that
yeah,
that
that's a
threshold.
very insightful, information because many synthetic biology product, develop, including metabolic engineering product, everything, tighter, production. I mean, I'm talking about chemical production, everything done beautifully, but their problem for final commercialization is something related to chemical engineering. Final purification is not sufficient
Right.
enough. Not necessarily, the production is perfectly done. Engineered bacteria produce tons of the whatever chemical, but final purification separation is not done well. But that's a typical, traditional chemical engineering problem, but they could not solve that problem. So I guess, I mean, some combination of different field and then, formulation is kind of important. One, pharmaceutical company know then for many, many years, but, synthetic biologists probably don't know, about that importance of that kind of thing. So we need to kind of rethink entire field to make more commercialized, more commercialization, commercialized kind of product in the market in the future, I guess.
That's right. And so that's a little bit maybe my personal vision, both of our group at BU and in the field, if I if I may have a view on it, is that I completely agree. So I really think it's these challenges at the interfaces, both of translation or sometimes materials and biology, where I think the challenges lie in the next 10, 20 years. And I think commercial entities will have an incredibly large role to play in that space. But I also think that in the basic science and academic science point of view, obviously we'll need to train people to be able to think across these different disciplines to tackle these challenges when they do go onto commercial companies. But I think there's also, it's like a shift in the way we look at the approaches. So our group is, our intention is to develop these like high throughput methods of experimentation and analysis that allow us to ask these questions, to even understand, like map what are all the materials and how do they impact this process? Because I think that's what we can do in the academic side, where company is like, okay, we just need one material to work. They don't have the time or money to like really map out the whole landscape. And so that's what we're hoping to do, both through the work I shared here today and some of these sort of application driven projects that we're working on now in our group. So hopefully.
Yeah. Thank you. I mean, I now, want to emphasize one more time the importance of collaboration. You beautifully show, I mean, you two different, group basically generate, huge impact, the outcome that is, Maria and you did great job. But I believe that in the future to solve a lot of problem, especially global problem, we do need the collaboration. And I believe that kind of generating huge impact, I think. So I guess, I mean, I really appreciate it. Can you demonstrate that on today? All right. I now
Thank you.
see,
Thank you.
yeah, 11 and 11 Central Time. I need to close. Okay. So thank you all for joining and staying today. We'll meet again on July 31st, Thursday, the same time, the same Zoom link. We'll have the Professor Irina Borodina at Technical University of Denmark and Dr. Wes Robertson at Medical Research Council Laboratory of Molecular Biology, UK. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with us. I will promote you to panelists who can speak and show your handsome and pretty face if you wish. Thanks. I stop recording.